Hey, how's it going, everyone? This is FixerMed. Welcome back to my high-yield biochemistry review series for the USMLE Step 1 NBME CBSE and NBME CAS examinations. This will be part 19 of my multi-part video series covering the entire discipline of biochemistry in a high-yield but comprehensive manner. So without further ado, let's go ahead and get started on today's content. Today, I'd just like to talk about the boring old lab techniques you need to know for this exam. So ensure that you have this memorized. Northern blot identifies RNA. Southern blot identifies DNA. Western blot identifies proteins. Southwestern blot identifies DNA binding proteins. And then ELISA, which stands for enzyme linked immunosorbent assay, detects antigen or direct ELISA or antibody when using indirect ELISA in patient samples. PCR, which stands for polymerase chain reaction, amplifies DNA fragments for replication. A good way to memorize the blots would be snowdrop. So snow would be southern, northern, ignore the O, W would be western, and then drop, D for DNA, R for RNA, ignore the O, P for proteins. Southwestern blot, you kind of just have to piece the two S and W together, so DNA and proteins. So that's how I like remembering it. It's always worked for me on the MCAT and NBME style exams. So with that out the way, we can go ahead and get started on questions. A research experiment shows that a nutrient causes an increase in gene products detectable by Western blotting, with no change observed in gene products detected by Northern blotting or PCR. What is the intracellular effect of this nutrient? Remember, snowdrop. Alright, think I gave you guys enough time here. If you need more time, please feel free to pause the video. Moving on now. The observation that the nutrient causes an increase in gene products detected by Western blotting indicates an effect at the protein level. Western blotting specifically identifies proteins, and the lack of changes in Northern blotting, RNA detection, and PCR, DNA amplification, suggests that the nutrient is not affecting transcription or DNA replication. Therefore, the correct answer is elevated protein translation. Translation is the process of synthesizing proteins from mRNA. Therefore, the correct answer is C, elevated protein translation. Let's go ahead and see why the other answer choices are incorrect. Increased DNA replication. This choice is incorrect because the experiment did not show any changes in gene products detected by PCR, which amplifies DNA fragments. Therefore, there is no evidence of an effect on DNA replication. Enhanced RNA transcription. This option is incorrect as there was no observed change in gene products detected by Northern blotting, which targets RNA. This rules out an effect at the transcriptional level. Augmented DNA methylation. This choice is not supported by the experimental findings as the focus was on changes in gene products detected by Western blotting, Northern blotting, and PCR, rather than alterations in DNA methylation. Upregulated RNA degradation. The experiment did not reveal any changes in gene products detected by Northern blotting, which identifies RNA. Therefore, there is no evidence to suggest an effect on RNA degradation in this context. Let's go ahead and move on to the next question. In a research experiment, a nutrient is found to cause increased gene products detected by Western blotting with no change observed in gene products detected by Northern blotting or PCR. What is the intercellular effect of this nutrient? Once again, snowdrop. Okay, I think I gave you guys enough time here. Pretty first order question. If you need more time, please feel free to pause the video. Moving on now. The experiment demonstrates an increase in gene products detected by Western blotting, which indicates a potential effect at the post-transcriptional or translational level. Since there was no observed change in gene products detected by Northern blotting RNA or PCR DNA, The focus is on processes occurring after transcription and DNA amplification. The correct answer, therefore, is augmented mRNA stability. Increased mRNA stability would result in a longer half-life of mRNA molecules, allowing for more efficient translation into proteins. Therefore, the correct answer is C, augmented mRNA stability. Let's go ahead and see why the other answer choices are incorrect. Accelerated RNA splicing. This choice is not supported by the experimental findings, as the changes in gene products were observed downstream of transcription, and there is no indication of altered RNA splicing. Enhanced DNA repair. The experiment did not show any changes in gene products detected by PCR, which focuses on DNA amplification. Therefore, there is no evidence of an effect on DNA repair. Elevated ribosomal biogenesis. The experiment did not provide any information about changes in ribosomal biogenesis. The focus was on gene products detected by Western blotting, Northern blotting, and PCR. Increased RNA polymerase activity. This option is not supported by the experimental data, as the lack of changes in gene products detected by Northern blotting suggests no alterations in RNA transcription. Let's go ahead and move on to the next question. In a research experiment, a nutrient is found to cause increased gene products detected by Western blotting with no change observed in gene products detected by Northern blotting or PCR. What is the intercellular effect of this nutrient? All right, I think I've beat Snowdrop into your guys' heads long enough. Let's go ahead and move on and see what the correct answer is. The correct answer is A, accelerated post-transcriptional modification. The experiment reveals an increase in gene products detected by Western blotting, suggesting a potential effect at the post-translational level. Since there were no observed changes in gene products detected by Northern blotting or PCR, the focus is on processes occurring after transcription and DNA amplification. Accelerated post-translational modification refers to alterations made to proteins after their synthesis, such as phosphorylation, glycosylation, or other modifications that can affect their function. As I said earlier, answer A, accelerated post-translational modification. Let's see why the other answer choices are incorrect. Altered chromatin structure. This choice is not supported by the experimental findings, as the changes in gene products were observed downstream of transcription and there was no indication of altered chromatin structure. Enhanced mRNA transcription. The experiment did not show any changes in gene products detected by Northern blotting, which focuses on RNA. Therefore, there is no evidence of an effect on mRNA transcription. Increased mRNA degradation. This option is not supported by the experimental data, as the lack of changes in gene products detected by Northern blotting suggests no alterations in RNA degradation. Augmented DNA replication. The experiment did not reveal any changes in gene products detected by PCR, which targets DNA amplification. Therefore, there is no evidence of an effect on DNA replication. And guys, that will do it all for this video. And as we approach the end of this high yield review series, I just want you guys to keep in mind the next review series you should probably look into is the genetics high yield review series, which at the time of this recording, I'm starting to make the slides for it. So biochemistry and genetics tie together really well. If you can move from one to the other, you should be solid. There's a lot of similar concepts that overlap. I'll be sure to make that high yield review series for you guys as soon as possible, since you guys have been liking the biochemistry and the general anatomy series. MSK should be coming out soon as well. General anatomy is great for students who have a good MSK understanding and just need an overview for the Step 1 CBSE and CAS exams. If you're using this as a first-time learning tool, it's not the best tool. Honestly, the high yield review series are not the best first-time learning tools. I'm working on making slides for first-time learners too, so be on the lookout for that as well. And anyways, this is FixerMed signing off. Make sure you have a good day and happy studying. And if I forgot to mention it, if you have any questions, comments, or concerns, please feel free to leave them in the comment section below. All right, goodbye, guys.